Hey 7th graders, today's assignment is the candy DNA molecule. Obviously you guys are watching this because you're not here today, so unfortunately you guys won't be building a real candy DNA molecule, but you guys will still be kind of following along with me to build one using my images on the next slide. So your learning target, I can build a model of DNA using candy. So we're going to go ahead and move on to this next slide. So this is a pretty short little assignment. So what we're using in class is Red Hots, marshmallows, and then gummy bears to show how to basically build a strand of DNA. And as we go through this, I want to kind of review and go through our notes that you guys added in yesterday with the DNA article summary. So let's go ahead and take a look at those. So what I kind of want to go down to right now is this little part that says DNA. So DNA stands for deoxyribonucleic acid. And our structure of DNA is what we call a double helix because there's two strands. Okay, just kind of like you guys were to take a ladder, you guys have two sides to your ladder. It's no different with DNA. Your DNA untwisted looks just like a ladder. Okay, you got one side here and you got one side here, and then you got the rungs connecting it. And those rungs in this case are going to be our nitrogen bases, our A's, T's, C's, and G's. Okay, so again, the difference between a ladder and DNA is obviously our little ladder twists. And you guys saw that when you guys built this the other day. Okay. If you guys unfold this, it kind of looks like that ladder where it has the two white sides. Those are your two sides of your ladder. And then the middle part are kind of your rungs. But again, in our case, that ladder twists up like a spiral staircase. Okay? It's made up of nucleotides. So if we were to talk about the building blocks of DNA, just kind of like we've talked about the building blocks of proteins being amino acids, or sugars and starches being glucose for the building block, okay? DNA is made of the building block of a nucleotide. So that's going to be one little part that we talk about when we do this model today, okay? So in that nucleotide, we're going to see 4 nitrogen bases. Again, those are going to kind of be our middle part of our ladder, those A's, T's, C's, and G's. A stands for adenine, G for guanine, T for thymine, and C for cytosine. And then on the outside, we have the white bars. Those are going to be our sugar phosphate backbone. And in that, you guys can see kind of this little pentagon shape right here, okay? That's going to be our deoxyribose sugar. And then you guys see this little, I like to call it the squished bug shape. Okay, that's going to be our phosphate group. So we see those alternating all the way down one side of our DNA. And since it's two-sided, we see it doing the same down the other side, okay? So we're going to go ahead and just start by building our sugar phosphate backbone. If you guys look right here on the key, we're going to see that our little sugar is going to be our marshmallow, and our phosphate is going to be our Red Hots. So I'm going to go ahead and click and drag a marshmallow, which is our sugar, our deoxyribose sugar, and then a Red Hot. That's going to be our phosphate. And what I'm going to do is I'm going to do about 5 of those alterations down the side. So go ahead and do about 5 marshmallows, 5 phosphates. That's going to be one side of our DNA. And then you guys are going to do the same thing for the second side of DNA. So make 2 strands for this. So you guys can go ahead, grab your sugars, which are your marshmallows, and grab your phosphates, which are your Red Hots. And I want you guys to do 1, 2, 3, 4, 5 down each side. So total, you guys should use 10 marshmallows and 10 Red Hots. So go ahead, pause this video for a second, and go ahead and build your sugar phosphate backbones for your 2 strands of DNA. All right. So you guys should have just gone through and completed your 2 strands of DNA, okay? And basically we need to connect these 2 strands now. So we need to add all those middle parts, or if we're again talking kind of about a ladder, we're going to add those rungs to the ladder, okay? So those rungs are going to be what we call our nitrogen bases. Again, we have 4 different nitrogen bases. We have adenine, which is our A's. We have thymine, which is our T's. We have cytosine, which is our G's. Our C's, and our guanine, which is our G's, okay? And we don't just randomly kind of put those in the middle all willy-nilly, okay? First of all, they attach to the sugar group. So anytime you guys are dragging one of your A's, T's, C's, or G's in here, you guys are going to make sure they're next to the marshmallow. They don't touch the phosphate. They actually just connect and bond with the marshmallow, okay? So let me grab a G, for instance. Here's a little orange G to represent guanine, okay? Now, we're not just going to put any gummy bear or any nitrogen base next to guanine or our G, okay? G's only ever pair with C's. They're kind of like best friends, okay? The way they fit together, it's like 2 puzzle pieces, and a G wouldn't fit with an A or a T, okay? So we're going to go ahead and grab its little best friend, which is our C over here, and we're going to pair those up, okay? Likewise, A's and T's are also best buds. So if I decide to put a T, which is a thymine, right next to my marshmallow here on our next part of our nucleotide, okay, the only thing I could put next to it is an A, okay? And the T's can go on either side, so you guys could have a T over here and an A over here. That's fine as long as they're always paired up. The crazy thing about DNA is there's not a specific pattern, so we're not going to go to a C and G next, and then an A and T, and then a C and G. Okay, the great thing about DNA is it can take any pattern. Okay, so I could put T, 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 T all the way down on this side, and then the rest it'll be A, okay? Or it could be all C's and G's, or it could be 2 more C's and G's, and then an AAT, and then 5 C's and G's. It's all very random, okay? And those randomness in our genes is kind of where we get different traits that make you different from everyone else. So when you guys are building this, try not to make a specific pattern as you guys go down. So what you guys are going to do to finish this is you're going to go ahead and add in your nitrogen bases and make sure that you guys pair them correctly. So remember that A's go with our T's and our C's and G's are best buds. We like to call those base pairs, okay? So think of that. That's really the only rule you guys have to follow, and that they go off of our sugar group here, okay? So go ahead, pause this video, take a second to finish adding your gummy bears or your nitrogen bases, your A's, T's, C's, and G's, to the middle of our 2 strands of DNA. Okay, now that you guys have finished adding and building your DNA strand, there's one thing that we want to label. So you guys will need to grab your draw tool for this. And what you guys are going to do is you guys are going to circle 1 nitrogen base or gummy bear, 1 phosphate group or red hot, and 1 deoxyribose sugar or what we call the marshmallow. So we're going to circle 1 of those. Okay, we talked earlier about nucleotides being the building block for DNA. Okay, this is 1 nucleotide. Okay, so we're going to go ahead, circle that, and then we're going to label it nucleotide. If you guys would rather go up to insert word box or text box, that might be a little bit easier for you. I have a mouse, so drawing isn't so bad. So there's 1 nucleotide, okay? That's what builds together. This is kind of like 1 puzzle piece that comes together to build DNA. Here's another one. Here's another one. And all the way down, we can look at all these nucleotides that come together to build DNA. But you guys just need to circle and label 1 nucleotide, okay? So once you guys have done with that, you guys have successfully built your strand of DNA out of your candy, okay? And you guys can go ahead and hand that in, and I will talk to you soon. Have a great day. Bye.